What if discovering a new life-saving medicine took a few short months instead of 15 years of lab work? For most of the 20th century, pharmaceutical research was a manual, physical grind, relying on years of trial and error experimentation. This timeline represents the modern baseline. Bringing one drug to market takes 15 years and an average investment of $2.6 billion. Despite that investment, the biological risk remains high. Over 90% of drug candidates fail during clinical trials, evaporating years of capital. Millions of patients wait for treatments because human-led trial and error has reached its physical speed limit. To bypass this bottleneck, deep learning algorithms shift discovery into digital simulations, analyzing vast genomic datasets to identify promising pathways in seconds. Generative models hallucinate novel molecular structures, proposing atomic combinations tailored to the specific geometry and needs of a disease. This purely computational approach replaces years of physical laboratory testing with high-speed in silico modeling. Software can now compress decades of chemical evolution into a fraction of a second. This capability is now facing its first major test in idiopathic pulmonary fibrosis, a deadly lung disease where patients often survive just 3 years. Insilico Medicine used its platform to identify a novel biological target called TNEC and simultaneously designed the specific molecule needed to inhibit it. The drug INSO-18-055 moved from its initial conceptual target to phase 1 human trials in under 30 months. This graph shows the clinical readout. Patients on a 60 mg dose saw a 98.4 milliliter increase in forced vital capacity at 12 weeks. While the placebo group continued to decline, patients on the AI therapy showed signs of the drug halting and reversing the scarring. INSO-18-055 represents a milestone in clinical validation. The algorithm identified the vulnerability, engineered the molecule, and demonstrated measurable benefit in humans. But an algorithm cannot physically manufacture the medicine it hallucinates. The industry built a new kind of hardware to keep up with the software. These are self-driving labs, where robotic automation integrates with AI to handle the physical synthesis and testing of new compounds. The discovery process revolves around the DMTA cycle. AI commands the design and analyze phases, calculating which molecules to test next. Robotic arms execute the make and test steps, feeding real-time data back to the AI in a continuous loop without human intervention. Human researchers have been elevated from manual lab work to conductors, managing the output of an autonomous discovery engine. Despite these gains in speed, a fundamental limitation remains: the computational biological translation app. As Demis Hassabis notes, biology is far too messy and complex to ever be encapsulated as a simple set of mathematical equations. Algorithms also struggle with the black box problem and data bias. Because proprietary pharmaceutical data is often incomplete or skewed, AI models frequently suggest molecules that are impossible to synthesize. This results in high-profile setbacks. Even highly hyped AI molecules like Exscientia's DSP-1181 and Recursion's REC-994 were ultimately discontinued after failing to confirm efficacy trends in clinical trials. A molecule can perform perfectly in a digital simulation or an animal model and still fail to account for the nonlinear complexity of a systemic human response. Artificial intelligence cannot eliminate the foundational risks of research and development. Human biology remains the ultimate filter, making software an accelerator for discovery, but not a guarantee of efficacy. The industry is currently in a massive learning phase, balancing the breakneck speed of computational science with the rigorous safety requirements of human trials. Regulators like the FDA are establishing new credibility frameworks to oversee these models, ensuring that speed does not compromise scientific integrity. If the industry can bridge the translation gap, the fusion of genomics and AI will allow for true personalized medicine, tailoring treatments to exact patient profiles. The ultimate question is whether cold algorithms can finally decode the warm, chaotic reality of the human heart.